Hi, today I would like to talk about the experiment number 6, diffusion and osmosis. Before we start discussing the topic, we should understand an important principle called homeostasis. What is homeostasis? It is a self-regulatory process by which biological systems tend to maintain stability while adjusting to conditions that are optimal for survival. Cells must move materials through plasma membranes into and out of the cytoplasm in order to maintain homeostasis. But how is this movement regulated? In fact, cellular membranes are selectively permeable because of their chemical structure. Cell membranes are phospholipid bilayers containing embedded proteins. Phospholipid fatty acids are hydrophobic and limit the movement of water. How can water pass through the membrane? Small amount of water can pass through the membrane freely by osmosis, and large amount of water can be transported across the membrane selectively by specialized protein channels called aquaporins. Generally, molecules pass through the membrane by one of the following ways. Most ions move through protein channels. However, larger molecules, such as carbohydrates, are carried by transport proteins via active or passive transport. What is the difference between active and passive transport? Active transport is a process by which materials move against a concentration gradient, and it requires transport proteins and energy. On the other hand, passive transport does not require energy. In this process, molecules moves along the concentration gradient in a passive process, and this includes diffusion and osmosis. But what is diffusion? And what is osmosis? Diffusion means the net movement of materials along a concentration gradient, from a region of high concentration to a region of low concentration. Molecules diffuse spontaneously as a result of their kinetic energy of random motion. On the other hand, osmosis is a type of diffusion that involves the net movement of the solvent from region of low solute concentration to region of high solute concentration, in the direction that tends to equalize the concentrations on the two sides. How can we demonstrate osmosis? We can bring a U-shaped tube with a selective permeable membrane and filled with water. When we add glucose in one side of the tube, water molecules will start moving toward the side where glucose is added, and we can observe the reduction in the water amount in the other side. What is tonicity? It is the relative solute concentrations in two solutions separated by a semi-permeable membrane. It determines the direction and extent of diffusion. A solution can be hypertonic, isotonic, or hypotonic. If solution 1 has higher solute concentration compared to solution 2, this means that solution 1 is hypertonic. If an animal cell is submerged in a hypertonic solution, water will leave the cell and the cell will be shriveled or crenated. If that cell was a plant cell, it will be plasmolysed, and the cell takes the appearance of a pincushion due to the plasmodesmata. If solution 1 has the same solute concentration as solution 2, this means that solution 1 is isotonic. If an animal cell is submerged in an isotonic solution, the net movement of water in and outside the cell is zero, and the cell will be normal. But if that cell was a plant cell, it will be flaccid. If the solute concentration in solution 1 is lower than that in solution 2, this means that solution 1 is hypotonic. If an animal cell is submerged in a hypotonic solution, the water molecules will keep entering into the cell, and the cell will be lysed or burst. But, if that cell was a plant cell, it will be normal or turgid or deplasmalized. The distance moved by molecules in a given amount of time is called the diffusion rate. It is affected by different factors, such as 1. Mass of particle, since high particle mass is accompanied with a low diffusion rate. 2. Temperature, since high temperature leads to higher diffusion rate.